My name is Alexander. I'm coming from the German Center for Diabetes Research in Germany, in the head office in Munich. And today I'll talk a little bit about our graph database and how we fight diabetes with graphs. Let's start with evolution. In the times of the dinosaurs and when there was no supermarket around, humans had the advantage to store energy when there was food, and they could store this energy as fat for the bad times. And this advantage turned out to be a disadvantage today, where we have supermarkets all over, and we have abundance of food, and we still eat a lot, and this fat got stored, and we got overweight and obese. And this obesity is a problem because this mainly causes diabetes. So what is diabetes? Diabetes is a metabolic disease where either insulin production in our pancreas is dramatically reduced, or the body poorly responds on insulin. Just to keep you in mind, insulin is the hormone, it's a protein that your body needs to get the sugar out of your bloodstream. And consequences of this sugar in your blood is that this sugar is less absorbed into your muscle cells or liver cells. The sugar stays in the blood vessels, and this is called hypoglycemia, and it's not stored in the cells, as I said. Diabetes has tremendous complications. I will come to that later, and currently it's not curable, it's only treatable. When we talk about diabetes, we mainly have the 4 different kinds of diabetes. Type 1 diabetes, come to that later. Type 2, gestational diabetes, which is diabetes at pregnant women. And we have some special forms that we are currently studying. On the right side, you see a picture here. So this is the healthy body. In your pancreas, there are some cells that produce insulin here, these little green dots. And this is your stomach, and you get your sugar into the blood vessels, and then insulin comes to it. And if they both come together, you get your sugar to the liver or to the muscle cells. And in type 1 diabetes, which is roughly 5% of the patients, these insulin-producing cells get destroyed by your own immune system. And this is independent from any lifestyle, and it's a gene defect. And people are dependent on an external insulin source throughout their lives. There are 20 genes, roughly 20 genes, that are involved in this process. And yeah, it's not curable. On the other hand, type 2 diabetes, roughly 90% to 95%, depending on the country. There is a little different picture. The cells are producing insulin, but there's an insulin resistance. So the production is still going on, but the body doesn't react on that. Normally, people don't realize that they suffer from diabetes. More than 150 genes are involved in this process, which increase the risk to get diabetes. And as I term it, the cocktail of evil is that you have a genetic predisposition, you have overweight, and physical inactivity. Just to show you some numbers here, worldwide we have more than 400 million people suffering from diabetes. It was thought that this was a disease from elder people, but actually 2/3 of the people are in their working age. We got another 200 million diabetics who are undiagnosed. This is very problematic thing. We have more than 1 million children suffering from diabetes. This is mostly type 1 diabetes. And 12% of all the healthcare costs are caused by diabetes. So it's a severe problem. We have to deal with this. When we go to the US, just to have some numbers here, 30 million are diabetes patients in the US. Every year there's 1.5 million coming to it. We got 85 million of pre-diabetes patients. And when we look at this number here down there, we got $90 billion per year that come from the reduced productivity of the people suffering from it. This is a chart which summarizes 1985 to 2009, so almost 20 years. And you see there the people that are obese here in the different states, and you see that this number is growing and growing, so even more people get diabetes and suffer from that. Complications, lots of complications. A cure on diabetes, so diabetes itself is not the real issue, but people are dying from the complications, namely stroke or heart attack. Like 1/3 of all the heart attacks is caused by diabetes. Nerves or blood vessels are degraded in your extremities, and some of you have maybe heard about the diabetic feet that have to be amputated for that. Of course, you get renal problems in your kidney. Diabetes is a complex disease. It's in your metabolism. Here's the pancreas, and it's a cellular process that is dependent on your environment or your lifestyle, but also on the genetics or epigenetics. I come to that in a second. And the problem on this is that on your environment and in your lifestyle, you inherit your lifestyle to the next generation. When we see this mice here, or these human, they are twins. Actually, they are genetically identical, but the epigenetics, which are these little labels here on the DNA, they are different on that level. And this causes diabetes, and this, on this level, you can inherit diabetes to the next generation. And a group of our researchers had some experiment in mice where you have different diets. So here, these mice get a normal diet, and these ones get a high-fat diet. And then you have an in vitro fertilization, and you see that the offspring, so the next generation, gets overweight, and with this overweight, the risk of diabetes is dramatically increased. So we are the German Center for Diabetes Research. We are a federal institute or organization that is academic and non-profit. We are funded by the Federal Ministry of Education and Research and by the states. We consist of 5 main partners depicted here in Munich, Tübingen, Düsseldorf, Berlin, Potsdam, and Dresden. We got some associated partners, and overall we got roughly 400 researchers from different disciplines, from basic research and from university hospitals. And our research areas are nutrition, we got patients in the clinics, we do prevention, we do genetics and cohort studies in basic research. And our main goal is to prevent diabetes, to treat diabetes, get new treatment for patients, and to prevent these complications that I showed you. Talking about prevention, we want to prevent diabetes in these subtypes, let's say type 1, type 2, or some other new types that we currently under investigation. And we want to identify them with some intelligent algorithms, and we want to treat them individualized. Now, how do we fight diabetes? So the typical researcher, it's everyday business. They are looking for information. They are looking for data within our organization, and this is like, looking for the needle in the haystack. And unfortunately, we got a lot of haystacks in the clinics, in basic research. And usually, these are easy questions like, what blood samples do we have, or what did we measure in the liver? And it turned out that these answers are not so easy to answer, so it's a complex query. And this originates because we usually were 3 different businesses, hospitals, as I said, basic research, and some bioinformaticians. And we all used to work for the same customer, the diabetic patient. But everybody of these researchers saw this diabetic patient a little bit differently. Some see it as an animal model, some look at microscopy pictures, some look on the metabolite, and so on and so forth. But now we have to look in a new way to our customer to combine different types of data. And that means that we have to share data. We have to look from as many perspectives as possible, and that means we have to connect the data together. There are two options. So one option would be to develop all the databases separately from each other, but this is not such a good idea. We want to build a new layer, so connected data. That's why we are here. And this is what we call DZD Connect. It's a Neo4j graph database. And now this graph database helps us, or helps our researchers, to answer biomedical questions. These questions can be answered across the locations. As I said, we have more than 10 different locations in Germany. We can answer questions across disciplines and across species. This database should be extendable, scalable, and this is one of the main features why we like graph databases so much. We have a visualization coming with it. When we have a look now to the data, we have homogeneous data here depicted in these similar icons. We have heterogeneous and homogeneous data. For the time being, we don't touch the raw data because of data security, and we classify this data at the different locations. We give them labels, and now we collect the labels so that we have the patient's data connected to each other. But in the end, of course, we reduce it to single labels, and this is how you see it's getting to a graph model. Why do we do graph databases at all? Well, the first answer is biology is connected anyway. So all the data in biology is connected. Our researchers like it because it's so easy to understand. The data model is like human readable, even for non-computer scientists. It's easy to query. I think it's much easier to query than normal SQL. It's scalable and adaptable. So when we have a new field of, or new discipline or a new location, we can easily adopt the graph model. We can extend it. And as I said, we have a very nice visualization. So these are the main reasons. When I talk about data, currently we include metadata. This is our preliminary data model here. A very big hub is an experiment because we are researchers. We do a lot of experiments. And in these experiments, we measure different parameters with different techniques or devices. In some of our experiments, we collect biosamples like blood, urine, liver, whatever you like. And we also include data from publications, from scientific peer-reviewed publications. And these all are connected in our data model. So examples for metadata can be, we have microscopic images. You can store the dimensions, what you see on the pictures. For parameters, we store the parameter name, the units, how it's measured. We have a biosample database where we store our biosamples on liquid nitrogen. And we want to extend this graph. So there are different possibilities how we can extend it. Other diseases, we can extend it by other diseases like cardiovascular diseases or Alzheimer's. There are a lot of literature databases publicly available. We will include electronic lab notebook data or protein database data. And here in the bottom, this is a special case where we enter the world of genes, proteins, and metabolites that are measured here in these experiments. And now we focus on that. This is a very nice poster by Roche. It's publicly available, and what you see here is all the metabolic pathways in your body summed up together. Now we are focusing just here on this blue box, and this is the zoom, and you see it's getting complicated. And if we zoom more in this, you see here that there are metabolites that are metabolized by enzymes, and they are regulated by other metabolites, and so on and so forth. And this database is very rich and very huge. And a startup company from the Helmer Center in Munich called Knowing Health, they dealt with this problem, and this is their data model, also Neo4j. And they connect these different worlds of genes, transcripts, and proteins. And their Neo4j database is very large, 800 million nodes and relationships. And we can include this database in our database. Another example how we extend our graph is Jan Krumsek here in the audience. He's now assistant professor at Cornell, and he's dealing with very rich data sources, and these data sources of metabolites are associated with phenotype data. In summary, we can say we build a metadatabase with graph technology, having different disciplines, different locations, and they are connected to each other. Now let's come to the examples. One example is that many of the medical doctors ask, so how many biosamples were acquired in visit 17 in our clinical study? This clinical study is called Prediabetes Lifestyle Intervention Study. And they are interested in these biosamples, and what did they measure on that? And this question is not so easy to answer. For this, we built a special data model. We have a clinical study here, and in this study, the patients come in different visits. And in these visits, the doctors make different experiments, and they measure parameters. And in some of the visits, they acquire some biosamples that they store and measure the data on that. And now our medical doctors can go to our Neo4j graph database. They can get their clinical study here, then they can extend it. They see the visits here, okay, and then they can browse to visit 17. And now you see that it's easily getting complicated. We have many different experiments here in pink, where we have the parameters in gray and the biosamples in yellow. And that's why it's so complicated to answer this question. Second example is something completely different. Here we want to connect data from different species, namely from human and our pre-diabetic pig model. And we ask the question, can human type 2 diabetes genes, can they be studied in our animal model? To answer this question, you have to know that all these areas here are different research areas where many people do research on that, and we have to connect them. You see here the data model, we connect them with knowing, and we have a set of genes here on top that are annotated for diabetes. These genes are mapped through the different databases, publicly available databases, into enzymes, and these enzymes metabolize different compounds. And so this is the human part of the data. Now we have the pre-diabetic pig, where we have a targeted metabolomics assay. We measure 16 different metabolites, and in the public database, these 16 metabolites are 31 identifiers, and then we have a union of these sets, and we come to the result that 7 compounds between the animal model and the human data could be identified. And now our researchers are looking into these metabolites in order to get new insights for diabetes. What's the outlook? We want to extend our graph by literature data that is publicly available. We have here peer-reviewed articles in a very large database called PubMed. There are, as I heard, 30 million texts in it, and about all the different basic research areas, and nobody can read all the texts anymore. And when you're looking for diabetes, you know, it's the bottleneck that you can read, or that you can read slowly. And we want to do an automatically learning algorithms on these texts here to learn gene names or to learn diseases. Here are some specific terms for it, high-fat diet, you see it. And in the end, we want to do that for many, many texts and see what are the connections between the texts. So every purple node here is a text that is analyzed by natural language processing procedure, by GraphAware, and then we see the overlap between different research texts. And we want to find these connections not only in the diabetes world, we also want to find connections between cardiovascular effects or Alzheimer's diseases, cancer, infectious diseases, and lung diseases. And these orange dots here are all organizations like ours, like the German Center for Diabetes Research in Germany. And we want to connect with each other to fight diabetes or to fight these other diseases. And this should summarize it again. So we have our people here that have different subtypes of diabetes or other diseases. We have the expert knowledge because we have the researchers here and the medical doctors in the clinics. We combine that now with graph technology, and we are on the way to better identify or cluster new subtypes of diabetes, to have an individualized treatment or to have an individualized prevention of them. And this should be a new center coming soon called the Digital Diabetes Prevention Center. We want to do pattern recognition on huge amounts of data that people from the society give us. We want to do supervised or unsupervised machine learning techniques in order to give back a benefit for the people who gave the data, and of course a benefit for the whole society. In my opinion, graph technology enables us to have a new level of diabetes prevention and treatment. We see events that we couldn't have seen with relational databases. And this is, again, because we can connect different disciplines, different locations, and different species. With this, I want to acknowledge the funders here, the Federal Ministry of Education and Research and the states. I want to thank all the scientists of the DZD, and I want to thank the organizers here and the organizer team for such a smooth organization and such a nice conference, and I'm happy to take questions. Yeah, so currently we are not storing regulations or like values. We are storing metadata, but in the future we want to do that. And well, I don't know if we store it in properties or in nodes. Not yet sure. I guess it's a performance issue. I would tend to put it in nodes. Yes. And because this comes from a biosample, you can link that to a biosample. I guess it makes sense to put it in a node, yes. Yeah, so there are many answers for that. So one is cipher queries. Second is I would, let's say like this, you have two nodes that you're interested in, like there's a gene in the animal model, and there's a gene in human, and you want to know if there's a connection between it. And so you have predefined graph algorithms. Find me the shortest path, find all the paths, find me a path with the length of, I don't know, 10 different other nodes. I guess there will be some algorithms solving that problem. Well, I sit there with the medical doctors, and we go through the data model. I mean, our graph is not so big currently. I mean, there are much bigger graphs out there. And we go through these different nodes, and we, you know, like on the fly, we say, can we answer these questions by this? Yeah, I guess it's a mixture between trying out and clicking and cipher and algorithms and training.
Hoping graph technology gets more, you know, more available to people in your communities.
Yeah, it's a tough question. You had a question.
Yeah, and your natural language processing.
Yeah.
On PubMed, for instance.
Yeah.
There was another tool that you said it was in the right-hand corner.
Yeah, by GraphAware. The guys that are out there having a booth. Well, these are the consultants. Correct me if I'm wrong. These are consultants on Neo4j technology. I used the publicly available version of it, and I analyzed some texts and looked to some connections between different kinds of literature data. Yeah, and it was pretty easy, I must say. Other questions? Yeah. Not yet. Other questions? Yeah. Say it again. So this project is only 18 months old. We had some prototypes, the one that I showed you with the metabolites between the pre-diabetic pig and human, and we have to investigate that now. Yeah, hopefully we have some new insights, but I'm sure the more data we import, the more we can answer, or more new insights or biomarkers we find. Yeah. Yeah.
Do you have currently, or do you have plans to provide access to these databases as subscription or whatever for researchers?
Not so easy to answer this question. First, this is available for our researchers, because it's the data that our organization has. Main problem here is that we have also patients' data, and we can't give that out. When we think about collaborations, and we're talking about academic collaborations, mostly of that, I guess it could be possible. There's a use and access committee dealing with this. So yes and no. Yes.
It looks like you deal more with type 2.
Yes.
Type 1?
Yes.
Is that just because it's 90% of diabetic?
I guess there's more research going on type 2, yeah, because there are more people affected to that, but we also do type 1 diabetes.
Are they related at all?
Well, I wouldn't say, I mean, they are related because it's somehow a metabolic disease, but these are not the not so many common genes to that. I would say it's different, yeah. And there are some new subtypes that we see. So we have obese or overweight people that are totally happy, don't have diabetes, and the other way around. So we have to see what's going on there to define new types or subtypes of diabetes.
What's your profession?
My profession, I'm bioinformatics.
Okay. So you're like subject matter expert?
Say it again?
Subject matter expert, right?
Yeah.
You're not IT person, you basically.
So I'm the interface, yeah. I'm understanding, or I hope I can understand our clinicians and our researchers, yeah, and to translate it into IT.
But you do have IT people who populate database.
The question was, so we have high-fat diet mouse and normal diet mouse, and when you in vitro fertilize them and bring them into the mother, the new generation gets fat. And this is because of epigenetics, this is because of your lifestyle and not about your genes. And the question was, is this model also true in human? And this is exactly what interests us. Are these the same metabolic pathways? Are these the same genes involved? Are these the same, let's say, lifestyle or environmental factors? So this is what we are currently investigating. Okay. Any more questions? Then thank you for your attention and have a nice afternoon.